Welcome friends, we are going to consider the DNA structure and its various forms. In this simplified structure, we can consider the double helical structure provided by the Watson-Crick model, in which the thickness of the DNA is 2 nanometer. The backbone that is clearly visible in the form of a spiral organization are basically made up of sugar and the phosphate alternatively, while the ladders or the steps of the ladder that are considered as the base pairs. In one base pair, there are the two bases which are bonded through hydrogen bonds. Now, the two bases of a pair are having the propeller twist. That means they are not the coplanar structure, rather they are having a tilt with respect to each other that strengthen their hydrogen bonding potential. The other important stability factors that we can consider covalent bond, that is in terms of the phosphodiester bond, and the most important one is the base stacking. The stacking of bases basically exclude the water in between of the nitrogenous base pairs, because the nitrogenous base pairs are hydrophobic in structure, so they won't love the water. So presence of water would disrupt the structure. So base stacking wouldn't allow the water, and thus it stabilizes the double helical structure. Moving to the next section, the various forms, in which the major forms we can consider A and B form of the DNA, which belongs to right-handed DNA, and this another one is the Z form of DNA, that is the example of left-handed DNA. The left-handed DNA is basically due to the alternative syn and anti-glycosidic bonds, while in the right form of the DNA, the glycosidic bonds are always in the orientation of anti-situation. Another important point we can consider the humidity, that means the water content when remains high along with the DNA, it assumes the B form. When the humidity is lowered, or you can say the salt concentration is increased, in that case it assumes the A form and Z form. Thank you so much. Stay blessed, stay safe. Greetings of the day. We are going to discuss the eukaryotic genome, its organization and regulation. In the brief video, we can consider the 4 different levels of eukaryotic genome organization. The DNA, which is of 2 nanometer diameter. When this DNA is wrapped around histone octamers made up of different types of histones, it assumes 10 nanometer fiber. Then again this 10 nanometer fiber assumes solenoid kind of a structure and turn into 30 nanometer fiber, famously known as chromatin. The chromatin makes the chromatin loop, and that finally organize into chromatid. And the two chromatids, when compacted properly, that is in the case of the metaphase and the anaphase, the metaphase chromosome can take the thickness of approximately 1400 nanometer. So this is the various levels of eukaryotic genome organization. It is the systematically packaging. That means whenever any part of the DNA that is having a gene is required, it opens up beautifully because of some sort of the chromatin remodeling. And that chromatin remodeling requires multiple kinds of histone modifications that occurs on the histone tails. The famous one or the major one are the acetylation, methylation and phosphorylation. The acetylation, that is caused by the histone acetyl transferase, especially it happens on the positively charged amino acids, for example lysine, and it causes the gene activation. If we consider the flip side, that means if we are going to consider the underacetylation, then it has been reported in the case of inactivation of the X chromosome that is turning into the bar body in the mammals. Similarly, the histone methyltransferases causes the methylation, and that especially at arginine and the lysine residues of the histone tails that cause the compaction, and that's why there would be no gene expression. The phosphorylation of the histone especially occurs at the serine and the histidines that is created by the kinases. It is having a crucial role in the regulation of cell cycle. So, thank you so much.